Great. Good morning, everyone. I'm going to talk about extraneous peaks and the assessment, especially in the context of compounded materials, and in particular, a method we use called chromatographic mapping. We do a lot of chromatographic analyses for stability, for batch release, etc. And so we clearly have to understand what all the peaks that we encounter as we assess and determine and elucidate potency or impurity profiles for our clients. So, before I got really into exactly how we do a lot of this work for a lot of our clients, I wanted to provide the regulatory standards that we use as a framework when we do interpretations around our chromatograms. So here are kind of the set of four that we use. The USP 1191, Stability Considerations in Dispensing, the 21 CFR, expiry dating, the current Good Manufacturing Practices Guidance, the most recent one that's been edited for 503Bs, which also provides you a reference document, ICH Guidance Q6A, Test Procedures and Acceptance Criteria per that title. The regulatory standard, the USP 1191, really talks about 5 categories of stability, of which I've focused on 2 here. The total count is chemistry, physical, microbiological, therapeutic, and toxicological. We deal a lot with the chemical, also known as strength, potency, activity, etc., and to ensure that the labeled amount that is being provided to us is within specification according to what our clients have defined as active and potent, typically 90 to 110 or slightly tighter. Toxicologically, to me, that translates, or we interpret that into no extraordinary formation of degradation products and a retention of potency, obviously, because often there's a trade-off. If you lose the active component, then you see an increase typically in some degradation components. So we want to make sure that we don't have an extraordinary increase. The CFR has guidance regarding expiration dating to include strength, identity, quality, and purity as components of characterization of the stability of that compounded sterile preparation. Purity, again, translates to us, we interpret that as no, you don't want to see an excessive amount of degradants or impurities during the shelf life or at the end of that expiration date you've established for that CSP. In the Good Manufacturing Practices Guide for 503Bs, there is a Table B, and I am focusing on sterile products. On line 1565, page 42, there is a table that describes all of the attributes that one should be looking for batch release and/or on stability. And in the very far end of that table, under Other Appropriate Specifications, it sends you to a small footnote at the bottom of the page, which is this. These include generally recognized attributes for each dosage form, such as those described in ICH Q6A or USP monographs. So for those of you who use the USP monographs for finished dosage forms, there are several monographs that have described organic impurities, any other individual impurities, total impurities, and that sort of thing. So I'll show you some examples of that after a bit. So if you go to this Q6A guidance, it provides a framework for what sorts of properties, attributes, and whatnot that specifications should be developed according to description, identification, assay, and impurities. And then it provides a decision tree which tells you if you do encounter a substantial amount of a degradant or an impurity, then it gives you a decision tree to how do you disposition that? What do you do with that peak and that kind of thing? So it provides some guidance on how to disposition that. This is the overarching paragraph. If you delve another few paragraphs, it actually has a section on parenteral drug products. pH sterility, endotoxin, particulate matter, extractables. In particular, for the generic industry, that typically involves the elastomeric closure, because a lot of those products are just glass vials, which are relatively inert, and the elastomeric stopper. Unfortunately, in the world of CSPs, the vast majority of products are contained in medical-grade plastic devices, syringes, cassettes, bags, all sorts of polyvinyl chloride, EVA, polyolefin, polypropylene. So a lot of these, it's an added complication for chromatography, because in standard analyses, a glass-contained product doesn't really leach into it many materials, but in CSPs, you see quite a variety. So, as I mentioned, there are several monographs in the USP that now declare, and I'm seeing more and more, levels of impurities. And I've provided 3 examples here: adenosine, ephedrine, and ropivacaine hydrochloride injections. And these have defined amounts and limits. And again, as someone mentioned yesterday, these limits are not only there at the beginning, but it cannot exceed these amounts at your expiry date either. So in terms of extraneous peaks within compounded products, we wanted to get into some of the meat of this presentation. And I wanted to describe kind of collectively the numerous sources that contribute to peaks in the chromatograms that we observe. The active pharmaceutical, it can, in itself, it has the main peak of interest, but it may have impurities. It may have some degradation. If you're using the powder material, it's typically more inert because it's in a dry form, but you'll have an impurity profile. For compounded source drugs that you get from generics, you'll often have some marginal amount of degradation that may have occurred depending on the age of that product as you source it and use it in your facility. Also, as you start to manipulate that, you're taking it out of its enclosed vial stopper situation and transferring it into plastic surfaces, the set, the EVA bag, or other container that you might use while you collect it and then redistribute it or collect it and dilute it and that sort of thing. So another parameter that we often see is excipient peaks. You need to understand and know where your excipient peaks are in your chromatograms. And excipients may also present with related substances. And benzyl alcohol is notorious, and I'll show you a chromatogram of just benzyl alcohol after a bit. And again, if benzyl alcohol is a preservative in the CSP that you're creating, it also may have components that grow or change during the aging of that product. And so you have to have a knowledge of that as well. And then you also may just have test method artifacts. This is, you have a diluent that you use to stabilize the samples, and it may have a large solvent front. You may use a large amount of an ion pairing agent in an analysis, and that also has a typical signature. And then, as mentioned, with all of the primary contact surfaces typically being medical-grade plastics, we do see a lot of extractables and leachables. And I just wanted to define for everybody that the extractable is when you introduce a solvent in contact with your materials and you see a pattern of peaks that come from that. Leachables are defined as those components that, as your CSP solution is in contact with that container, it delivers materials into that CSP. And so the consequences are that there are peaks that are derived from these materials that do appear and are encountered in chromatograms. And in fact, we have seen that the longer that CSP is in contact with that surface over your 30, 60, 90, 120 days, those particular peaks may grow in peak area count. So we use in our shop a chromatographic mapping approach. In other words, we characterize independently the contribution of each of the components, and then we collectively, by definition of relative retention time, understand and assign those peaks within the chromatograms. And I wanted to give some real-life examples of some of the materials or information and idiosyncrasies from our methods that we have a knowledge of. Here I have remifentanil and succinylcholine. One has a very unique diluent, and you see a large solvent front. The other, very high ionic pairing agent, and you also see materials there. So active pharmaceutical ingredients. Another contribution is their related substances. Adenosine has a variety of related substances, as you can see, and they're all well resolved, but you need to understand what they are and where they're at. The ropivacaine actually requires two independently separate chromatographic methods because related compound A chromatographs differently than the enantiomeric related compound B. And so you have to understand and know where those are at. And then succinylmonocholine is also has the monocholine versus the succinyl in it, and you have to understand where that is as well. Excipient peaks. So we run the excipients, and we do run them for selectivity, so we understand where they elute in the chromatograms. Rocuronium has a sodium acetate, sodium chloride matrix, and so we understand where that peak is. And in fact, ephedrine, which we run on a Hillock system, the sodium chloride interacts, and we actually do see an assignable peak in the chromatogram for that diluent. So the benzyl alcohol, this is benzyl alcohol unaged, and it has at least 4 or 5 additional peaks that are associated with it. And so if you were to run a product that contained that, you really need to understand how to assign those peaks to the ingredient and not degradants of the API that you're looking at. Methylparaben comes out earlier compared to lidocaine if you're using preserved lidocaine. Extractables, leachables, again, the top chromatographic profiles are demonstrating in the absence of the active component, you see peaks that are related to exposure of the diluent to the interior of a syringe, in this case, for the fentanyl and the oxytocin. This is highly exaggerated for very dilute solutions that you have to directly inject for analysis. Those peaks will be of some consequence versus those samples which you're able to dilute, and therefore, your interferences are reduced by the fact that you're diluted several-fold. So in order to collectively know what these elements are, as you go to ultimately monitor something on stability and understand what those results are telling you, you have to understand what's the known behavior of the CSP. What are the impurities in the API or the source drug you're using? How does that, during method validation, you run very stressful conditions. High acid, high base, heat, peroxide, light. So you have profiles in your validation reports that should indicate under said condition, I'm seeing a profile where I have 2 or 3 additional degradant peaks. And so you should have that knowledge available to yourself when you're running your stability studies. So you should know where your contributions from method artifacts are, your excipients, if the excipient has related peaks or degradants associated with it, the container closure, extractables, and leachables. So the other issue is that the extractable leachable profile is specific to the container, the CSP solution, and the method that you're running. Every method has a different elution profile for every analyte you're running, and those extractables, leachables will elute in different regions and different methods. So collectively, this is how we pull this together. We have an extractable, leachable profile for glycopyrrolate. We have the benzyl alcohol component, and then we have the stability sample, and we know where the glycopyrrolate comes out, and therefore there are only two unaccounted for peaks in that chromatogram that we can actually assign through PDA detection as well, that they are truly glycopyrrolate related. They're not container related, they're not benzyl alcohol related. The way in which we collect a lot of this information to be able to do this is that we know where our test method artifacts and idiosyncrasies are. We capture a lot of that information in our methods to ensure that the analysts that run these stability studies on a day-to-day basis know and understand the method that they're running and are familiar with all the potential properties or peaks that they might see. If it's the active pharmaceutical ingredient or the source drug, we often run samples of those on the methods to determine if there's any consistent peaks that are present at the start of any study. So we can attribute that either to the API or the source drug. And in most cases, they're impurities, and often they don't grow. They're just synthetic byproducts that couldn't be isolated away from the API or are present also then in the source drug. And to utilize your force degradation profiles, which I mentioned a few minutes ago. If it's your excipient peaks, know where those elute. Label them in your chromatogram and your representative chromatograms in your methods, so your analysts won't get confused. We do a lot of molecules, and so they really need to understand what excipient goes with what product. And then, if in fact your excipient has related substances and maybe it does degrade over time, your best option to collect that is at the beginning of your stability study, run a side-by-side placebo solution that contains all the contact surfaces, same device, everything except the absence of the active component, so you can determine that it's either an extractable or leachable, or it's related to the benzyl alcohol or the ingredient. Citric acid is another one that pulls things out too. And again, just make sure all of the collective and the anecdotal and the analytical information is contained in your method, so we do in our case, our analysts are very familiar with what they're seeing. If in fact you want to characterize independently your extractables and leachables, there are several techniques to do that, one of which is monitor the placebo solution. Also, you can run an extractable experiment where you expose the container of interest to saline, low pH water, high pH water, maybe a solvent water mixture, maybe elevate the temperature to kind of get your worst case scenario, and then chromatograph those solutions on your standard methodology and determine where that particular profile is for those extractables from that particular container. And just to let you know that we use also one further guide regarding FDA's thinking on extractables and leachables, whereby they're saying that anything less than 1 microgram per mil in your solutions is typically acceptable. Above that, you do need to start evaluating what that is, where it came from, that sort of thing. So that's kind of the threshold we use when determining whether we need to tell a client, well, perhaps we need to evaluate what exactly might be coming from the container closure. So.
That was a very nice overview of the use of chromatography. What I'm interested in knowing is two things. Today we deal with a lot of these compounds that are known, USP monograph articles. What I'm interested in, though, are the 2 or more that are brought together in a container closure system where the stability profile isn't known and maybe the reference material for the associated degradants aren't available. How do you solve that problem? Or what's your proposal for solving that problem?
Well, we typically, for example, a cane fentanyl combination. So we'll run force deg on that combination. So we'll get an idea of what that profile looks as the 2 components are present together. We also historically will do them individually combined, so that we have a sense of what's coming from the fentanyl, what's coming from the bupivacaine, and so we can understand, that scenario.
And in the case where you don't have UV activity or strong enough UV activity, do you or have you developed other detection methods, mass spec, ICP, or other methodologies in association with hyphenated techniques with the chromatography?
We have not typically do orthogonal testing. If there is something that's truly different, then we will actually outsource a full-blown extractable leachable study or some other component. Or we have multiple technologies, but typically we haven't done anything that bizarre that is outside the realm of what you're referring to.
Yeah, that's good to know. Well, thank you. That was a very informative presentation, Lisa.
Thank you.
So, Lisa, I actually had a question.
Sure.
When you were discussing doing a reference formula without the active ingredient in there, right? So what I do is I'm a 503A, 795 only, no sterile products, and I need to validate oral suspensions that are in things like Aura Plus and Aura Sweet. And we're using, per the FDA guidance, we're using the finished product as our starting material. So in other words, tablets and capsules that contain multiple inactive ingredients. And so there's no way for me to get, when we're compounding the finished product, it has lots of excipients in there from that, and we can't, but we want to know the baclofen, for example, out of the tablet. So how would we go about figuring out how to see through all the junk to find the API in a situation like that?
Well, unfortunately, tablets don't declare the exact concentration of each component, but it does identify each component. So you would have to, we'd have to, purchase each excipient, run it against a proposed method, and see where that excipient may interfere or not interfere. That's how we would kind of deconvolute the active component against each of the excipients. In sterile products,
Sure.
your preparation is defined. It tells you exactly how much of everything you have in there. But for oral or liquid dosage forms, you just have to kind of deconvolute based on the composition, collection of each of the excipients, and then determining, hopefully that there's no interference, or if you have to tweak your method or modify it around that parameter, then you need to do that.
Okay. Thanks, that makes a lot of sense. Thank you.
Okay, great.
Okay, thank you very much, Lisa. I appreciate that. Thanks. Very good information.